Welcome to the second Synapse Session. Essentially, the mission of the awareness team is to raise awareness about mental health and neurological disorders. So, yeah. Okay. And unfortunately, this is a rerun through of the slides as we experience some technological difficulties in the actual second Synapse session. So for these interactive questions, we'll just be skipping over them for the sake of time. Okay, so um, I'm very excited to introduce Ms. Jyoti Chaurasia. She is a PhD student at the National Center for Cell Science, Pune, India, a premier research institute for cell biology research. She is currently studying ALS, otherwise known as amyotrophic lateral sclerosis, a fatal motor neuron disease. Her work explores the mechanisms of the spread of infectious proteins among brain cells, ultimately leading to neurodegeneration. She has been keenly interested in neuroscience since the start of her graduation and post-graduation in biochemistry. Other than academics, she loves traveling and exploring culinary dishes of India. So her email is also included on this slide. So if you have any questions, feel free to reach out to Miss Jyoti via that email address. Okay, so now I'll be passing the microphone to Miss Jyoti. Um, I will also,
Thank you, Danielle. I'll present from my slides.
yes, I will give you hosting permission, so you should be able to share.
Okay. Is the screen visible?
Yes, we can see it.
Okay. So, hello everyone. My name is Jyoti Chaurasia, and I'll be presenting the following paper from Diana Arseny and Benjamin Risque de Falcon. So this paper is a follow-up of follow-up of the study that the same group did for TDP-43 in the previous years, where they have discovered the structure of FTLD TDP type B and ALS. So they in this paper they actually used the cryo-electron microscopy to find the TDP-43 fold in FTLD type A. So the group actually is from MRC Laboratory, University of Cambridge, uh, where Dinah Arseny explores the mechanisms of brain health, aging, and the disease, specifically including the lysosomal proteins. Whereas Benjamin explores the molecular basis of protein assembly in amyloids in various neurodegenerative diseases. So, about the disease, FTLD is frontotemporal lobar degeneration, which actually occurs through tau or TDP immunoreactive pathology. However, many other proteins to be involved are reported, but tau and TDP form the major part of the disease spectrum. It is actually a heterogeneous clinical syndrome where people experience progressive changes in behavior and language and also in personality. So the major clinical subtypes include the behavioral variant where there's a change in behavior, and semantic and progressive aphasia variant where there's a change of or disabilities in language, while when there is a motor involvement that comes under the umbrella of ALS. So various genes mutations are, give me a minute, I'll just, various gene mutations act as a risk factors for FTLD. Sorry. Various gene mutations act as a risk factors for FTLD in both symmetrical and asymmetrical atrophy of brain, where today in this paper we are focusing specifically on granulin mutations. Granulin is a gene which produces progranulin, a synapse-related protein, which functions specifically on a synaptic terminus. And mutations in GRN genes actually cause abnormal aggregation of TDP43 aggregates. How actually this is connected is still elusive. So we can say that the aggregation of misfolded and dysfunctional protein is specifically the pathological hallmark in this disease. And from the statistics and from the cases that have been reported till now, tau and TDP are the predominant protein aggregates, specifically in the neurons, in the cases of FTLD and TDP. So this is the immunohistochemistry images of all the types of and subtypes of FTLD, which is caused by tau and TDP. FTLD TDP is the type we will be focusing today, and it has specifically 5 subtypes, including ALS. So the arrows actually indicate the aggregates specifically found in the various regions of the brain tissue. This is from Irwin et al. Acta Neuropathologica. So FTLD TDP is characterized by aggregation of the TDP43 protein, as we saw previously. And the subtypes include the 4 major subtypes, where type A has dystrophic neurites and neuronal cytoplasmic inclusions, which are short. And it is generally caused by granulin mutations and the mutations in the chromosome 9. While type B has few dystrophic neurites, but more neurocytoplasmic inclusion. Neuronal cytoplasmic inclusion include the cytoplasmic inclusions which are formed in the cytoplasm of the cell. Because TDP43 is a nuclear protein. So when it comes out during the disease in the cell, it actually aggregates. And that is what the major problem arises. So following with the next type is the type C, where the dystrophic neurites are longer, while the neurocytoplasmic inclusions are very less. And type B has dystrophic neurites and neurocytoplasmic inclusions along with neuronal intranuclear inclusions. These are the inclusions which are formed in the nucleus. And this type D is also associated with various clinical subtypes. So as we can see that in the disease, specifically in the neurodegeneration, protein morphology actually matters the most. So the major question arises that why does the protein morphology matters? So protein morphology means that that is the 3D shape of the protein, which is central to its function and specificity and stability. So we actually what happens is that the one-dimensional gene and code actually determine the linear amino acid sequences, and the side chains of amino acids give it the proteins, its unique properties that affect the folding of the proteins. So what actually happens is this folding process is governed by thermodynamic and kinetic principles. One of the explored principle is the Levinethal paradox, which actually says that protein cannot possibly have all the conformations randomly, but still there is a paradox about how it actually manages to reach its lowermost energy state in such a short time. So now this morphology actually dictates the properties which determine the biological outcome. And when these are altered, the neurodegeneration, specifically the neuronal proteins, when the morphology and the inherent properties of these proteins alter, neurodegeneration specifically is seen to be occurring. So these are the secondary structures of these are the secondary structures, primary, and the following more motifs, domains, and tertiary structures of how different proteins actually form. Alpha helix is commonly observed in most of the amyloid fragments that are found in various neurodegenerative diseases that I have highlighted. So talking about the protein. TDP43 is a RNA binding protein, that is a nuclear binding protein, which has major roles in RNA processing. By major role, I mean it majorly focuses on RNA processing mechanisms, but however, it is also involved in mitochondrial uh, fusion and fusion machinery, autophagy uh, related mechanisms, and also in ubiquitin pathway. So it's a but its major role is actually related with RNA processing. So the structure actually contains the N-terminal domain, which is specifically whose specifically role is for oligomerization, and two RNA binding motifs, which regulate all the RNA related mechanisms. One more domain is the C-terminal domain, which is also known as low complexity domain. It is specifically it is specifically utilized for protein-protein interactions, phase separation, and it is prion-like. So, and this prion-like property is actually the major cause of aberrant aggregation in the diseases. So, what happens is that since it is involved in ubiquitination, so the cytoplasmic inclusions which are formed in diseases usually have dominant mutations, and the inclusions are ubiquitinated. So in ALS and FTD, what happens is that when there is a mutation or some other factors that are being thought to be as risk, so when it happens, the nuclear TDP43 comes out of the nucleus and forms cytoplasmic inclusions in the cytoplasm. So this disrupts the key RNA processing events, and there is an inclusion of other unwanted cryptic exons which hinder the normal functioning of the neuronal proteins. Now the question arises how this TDP43 actually became the therapeutic target of neurodegenerative diseases. So this was identified in 1995 in HIV gene and was called a DNA binding protein. And in next following 5 years, it was came to know that it forms UG and TG rich sequences. It binds to these kind of sequences, and it is involved specifically in RNA splicing and transcription. By the following years, the cytoplasmic ubiquitinated inclusions were found, and notable discoveries were occurred, reporting it as a therapeutic target in the neurodegenerative diseases. So the healthy nuclear TDP43 is involved in mRNA splicing, nucleoplasmic shuttling, mRNA transport, miRNA biogenesis, translation, and stressed granule formation, along with transcription. It is also involved with other organelles that is not highlighted here. This was taken from Bazida 2023. So this is the difference. This is taken from the same paper as it was mentioned in the previous slide. This is the degenerative effect that is caused when there is a loss of function in the nuclear TDP and gain of function in the cytoplasmic TDP. So what happens is when there is a loss of nuclear function, there is a deficient in RNA granule transport, loss of axonal transport functions, which actually cause degenerative effects in the neuron. And this actually happens specifically in the motor neuron. So as I discussed previously, that the group actually explored the cryo electron microscopy structures of TDP43 and found a new type of fold in the brains of 3 individuals. Okay. So the approach was, as I mentioned before, that the group showed TDP43 assemblies in type B, FTLD TDP. The other type, that is the type A, was most common variant, but the structure was unknown. So the group utilized an integrative approach using cryo-EM and mass spectrometry to identify the disease-specific fold and modifications that modulate the fold. So this is the cryo-EM images of what they found during the initial studies of the amyloid fold. So before moving further, what is cryo-electron microscopy? So it is similar to electron microscopy, but the sample is being studied in cryogenic temperature, and this cryogenic temperature allows us to study the sample in the native stage. So what we do is that we rapidly freeze the sample on the electron microscopy grid in Virch's ice, and we automate the 3D resolution in the images inside the ice crystal. So it is specifically the transmission electron microscope with a special specimen holder that enables us to view the frozen samples. The frozen or the freezing temperature is mainly maintained by liquid nitrogen, helium, and this is what is the workflow of making the sample for cryo-EM. What we do is we take very less amount of samples at around 2 to 3 microliter, and it is plunged in the and this EM grid is washed and prepared, and then it is plunged in the liquid ethylene or nitrogen or helium, any of the cryo temperatures. So this is how the protein actually settles or sets on the EM grid with the carbon support. So this slide actually slide is taken from a different paper, but it actually shows the workflow of what the group has done in their paper. What we do is that we prepare samples, and we do the cryoimaging of the sample, and followed by classification, sorting, building the initial maps that actually helps to build the protein fold, and we do the model building followed by interaction and complex identification. This workflow actually helps in the refinement of the model that the person will be developing, followed by to understand how the protein is affected by the modifications. For that, usually mass spectrometry is done. So the sample collection and extraction process was done in such a way that the group actually checked for neuronal cytoplasmic inclusion, short DNs, in the patients of type A FTLD, and there initially 5 patients were taken, and the prefrontal cortex tissue was used for immunohistochemistry analysis, and these are the, if I would zoom, these are the aggregates that were found in most of the individuals prefrontal cortex tissue. So out of 5, 4 were with granulin mutation and 1 was with wild type granulin, and they followed the study with one patient with wild type and 2 granulin mutations. So the TDP43 inclusions were extracted with sarcosyle method that they have previously used in FTLD type B. Following cryo-EM studies, the group found that there was 10 to 15 nanometer wide straight granular surface filaments, which were confirmed to be TDP43 filaments, specifically in the C-terminal fragments with immunogold labeling. So immunogold labeling is a technique that is used to visualize the surface molecules using specificity of antigen-antibody reaction, using colloidal gold particles or a heavy metal stain. Specifically, uranyl formate or uranyl acetate is used. This was followed by helical reconstruction of cryo-EM images, which was of 2.4 angstrom resolution, and the group found that the single amyloid protofilament was stacked with TDP43 molecule in a new characteristic fold. And since this amyloid fold was unknown previously, structural analysis of the fold was followed. So in the structural analysis, which enabled the researchers to view the peptide group's ordered solvent. They developed with an accurate model of TDP43 fold, which was found to be with right-handed helical twist. They called it a chevron badge fold since it was composed with five stacked beta layers of LCD domain, that is the C terminal domain I mentioned before, and it was distinct from the previous fold that they observed in ALS or type B FTLD. The type TDP TDP type A had glycine rich region and hydrophobic region. This is the pink one, the glycine rich region, followed by an white colored hydrophobic region. And the third and fourth layer were QN rich. And there were, in the outside of the fold, there were RRM regions which formed the part of the fuzzy coat of the amyloid fragment. So this is the comparative fold with the chevron badge that is being usually used in the military thing. So the fold was found to be centered and kinked with 11 residue beta strand starting from A321 to Q331, which formed steric zippers in the neighboring of third to fifth layers, with imparting its resemblance with the 3-bar chevron fold. Oops. And these are the secondary structures of type A FTLD fold that showed that the single TDP43 molecule perpendicular to the helical axis, this was with 5 layers, has been shown. Now, following the secondary structure development, the group also found that there were some conformations with alternative conformations. Okay. Specifically, there were alternative conformations in N-terminal domain and in the layer 4th and 5th with 2.5 angstrom resolutions. So what they found is that in the N-terminal domain, G281 and G295 actually followed the same path as residue R272 and P288 in the main conformations. This exposed the R293, here you can see, outside from the fuzzy coat. In the diagram, there is the blue one is the original main conformation, while the yellow and pink one show the alternative conformation in both the areas, which was found in 5% of the folds. While in the alternative conformation that link the fourth and fifth layers, M336, A341, and Q344 were buried, and M339, S342 were surface exposed. This shows the mapping of the filament segments with locations of micrographs that revealed that the TDP43 molecules could have different structural variation coexisting with the main conformation in the individual filaments. So from this finding, the researchers confirmed that the amyloid filaments do not always have uniform repetitive structures, but they also show the structural variability within the filaments. And they also speculated that this can be exploited in the development of diagnostics and therapeutics against these kinds of neurodegenerative diseases. This was followed with mass spec to identify any modifications that affected the formation and the functioning of the, or maybe the disease causing property of the fold. So they found that the arginine 293, which was exposed in the alternative conformations, was specifically located on the RG motif of TDP43 LCD. And this actually participated in the functional interaction with RNA binding proteins. And it was regulated by citronylation and methylation. So the group hypothesized that the burial of this residue may have facilitated by PTM, specifically citrullination, in which the charged guanidinium group was hydrolyzed, yielding a neutral uridyl group. So this explains the chemical reactions that would have caused specifically the post-translational modification. As the side chain of the R293 was completely buried in the main conformation and was partially compensated with hydrogen bonds. So the overall effects of this buried charge were destabilizing. So specifically two types of modifications were found. So analysis for citronellation, the group analyzed the brains of the patients with FTLD type A by mass spectrometry and found citronellated TDP-43 molecules specifically in the arginine 293. So they speculated that this facilitated the formation of TDP-43 filaments by removing the destabilizing charge on arginine 293. And arginine 293 was also found monomethylated specifically in the alternative conformations where the R293 was surface exposed, specifically in the minority TDP-43 molecules. So, and in the previous studies, this monomethylation has been also reported to reduce the TDP-43 assembly in vitro. So, the group concluded that these post-translational modifications influence the local structural variation of TDP-43 fold by affecting their charge and size. These were the representative mass spectra of the TDP-43 in which the citron-related arginine 293 is shown and monomethylated spectra is shown. This is specifically for individual 1. So at the end, they compared the FTLD type A chevron fold with the double spiral fold of FTLD type B. So they found was that the N-terminal truncation site is here shown with the scissor symbol, and R293 is indicated with blue dot. So they found that this chevron fold was having the same part of TDP-43 LCD, but this chevron fold had 10 extra amino acids specifically of the N-terminal domain. So the major findings that the group actually found was that TDP-43, okay, the meeting is... So the TDP-43 amyloids in TDP for FTLD type A and type B were different, and that may characterize the different neurodegenerative conditions that we observe in patients. And we also explored the roles of post-translational modifications, specifically on arginine, that actually affected in filament formations and induced variation in the individual filaments of structures. So the group has also mentioned that in the future studies of mechanistics of this TDP-43 assembly can help us to diagnose and find therapeutic compounds that actually target these amyloid folds of TDP-43 in future. Thank you.
Thank you so much, Ms. Jyoti, for your valuable insights into the paper. Now, just to kind of conclude, I'm going to give like a very quick overview of the paper. So I will share my screen and okay. Okay, starting off with the abstract, essentially the main topic of the research article is a protein called TDP-43, which stands for TAR DNA binding protein 43, which abnormally accumulates in most of ALS and about half of FTLD cases. So this is a result of mutations in the TAR DBP gene, which encodes for TDP-43. So there are 4 subtypes of FTLD TDP ranging from type A to type D, where each has different patterns of TDP-43 accumulation. However, the structure of TDP-43 in type A was unknown, so this is the essential research gap the paper is addressing. As a result, the researchers utilized cryo-electron microscopy to study the filaments of TDP-43 from the brains of 3 individuals that had type A FTLD-TDP. They discovered that the protein formed a new type of amyloid filament fold that resembles a chevron badge in all 3 individuals. The term chevron originates from the V-shaped insignia that is worn on military or police uniforms. This is significant because it was distinct from the double spiral fold seen in ALS and type B FTLD-TDP. These findings support the idea in which different neurodegenerative diseases are associated with unique protein structures. So kind of giving a little bit of a deeper rundown of the methodology, human tissue samples were acquired from the brain library of the dementia lab at Indiana University School of Medicine. The chosen samples had to follow certain criteria, such as having a lack of comorbid neurodegenerative pathologies. The clinical presentations of the individuals included non-fluent variant primary progressive aphasia and behavioral variant frontotemporal dementia, in which these clinical presentations had been previously associated with type A FTLD-TDP. Since most mutations that cause diseases are found in protein coding regions of the genome called exons, whole exome sequencing was conducted to find genetic variants linked to FTLDs, especially with the GRN gene, which is commonly mutated in type A FTLD-TDP. A polymerase chain reaction, abbreviated as PCR technique, was used to detect repeat expansions in DNA, since ALS and FTLD can be caused by expanded repeats in genes like in chromosome 9. TDP-43 was extracted from the prefrontal cortex using a Sarcosyl-based protocol. Sarcosyl is a detergent that helps separate soluble proteins from insoluble aggregates, such as the misfolded TDP-43. Centrifugation and sonication, which is essentially using ultrasonic waves to break up protein clumps, were then utilized to isolate the sarcosyl insoluble TDP-43 aggregates. Some other aspects of methodology include the histology. So histology was utilized to visualize proteins in the tissue sections using antibodies and locate TDP-43 aggregates in brain tissue under a microscope. This helped confirm that TDP-43 was located in the expected brain regions. Immunoblotting was also used to detect specific proteins and verify the molecular identity of the extracted proteins. TDP-43 elements were digested into smaller peptides using enzymes. They were analyzed using mass spectrometry that can detect post-translational modifications, which are changes made to proteins after they are produced. The two identified post-translational modifications were citrullination and monomethylation, both of which result in unique filament structure that is found in type A FTLD-TDP. These filaments were then imaged under cryomicroscopy. The researchers used various software platforms such as Chimera X to construct 3D models of the filaments at the atomic level. These structures will help design targeted therapies and compare structural differences among these disease subtypes. And then also, as we start to look at the limits and biases of the research, you may want to consider how there was a limited sample size, so their findings may not accurately depict the biological variability across a bigger population. In addition, the samples were derived from post-mortem brain tissue, so the results could have been affected by tissue degradation and end-stage disease changes. And then going on to some final reminders. Recordings of the Synapse sessions will be recorded and uploaded to Simply Neuroscience's YouTube channel. If you're interested in attending future Synapse sessions, please subscribe to SN's newsletter. And if you're interested in becoming a guest speaker at future Synapse sessions, there is an interest form that you can fill out. And if you're interested in receiving a certificate of participation, you'll need to attend at least 3 sessions and submit some brief reflections. And so you can visit the Google form for more information. And I will be passing the microphone to Miss Jyoti to give us some final remarks.
Thank you, Danielle. So, as a final remark, I would say that you guys can explore in the neuroscience field before being like before entering in a PhD if you are planning to, because if we exclude neurodegeneration, this field is very vast. And if you can explore some interdisciplinary research, so it would be very better for you in a PhD or in your research career, because interdisciplinary research has been way more effective as compared to doing a monotonous work. So I hope you guys have a good time exploring the field of neuroscience.
Thank you so much. I agree. Neurodegenerative disease research is certainly very interesting and also very important, especially nowadays. So thank you so much for watching this recording of the second Synapse session. And if you have any suggestions for topics at future meetings, feel free to email me or Mariia. We are the co-directors of the Awareness Department at Simply Neuroscience, and we would be more than happy to incorporate your ideas. Thank you.